Hi everyone. Let's start talking about the specific genes involved in familial Alzheimer's disease. The best place to start telling this story is with a gene named APP, which is the short name for amyloid beta A4 precursor protein. In order to tell the story of APP, it's necessary to discuss what amyloid is. Now, the word amyloid can basically be thought of as meaning starch-like. And it refers in this case to the fact that if you section a tissue, a brain, and then you stain it in a particular way with chemicals that will reveal certain structures, if you stain it with something that reveals starch-like substances, then in the case of someone with Alzheimer's disease, that will often reveal things called amyloid plaques. Now, the amyloid plaques were first defined by Alois Alzheimer a bit over 100 years ago now. The history of this is actually discussed in this essay here from 2006 in Current Biology by Ralph Dahm. And in that paper, you can find a reprint of one of Alois Alzheimer's drawings, which was a section from the brain of a patient of his with senile dementia, which he then stained in a particular way. And when he stained this particular brain section, he saw something called neurofibrillary tangles, which we'll talk about later. They can actually be seen here. It's kind of like a comet-like stream away from a nucleus here. But he also saw these blobs that were staining for something starch-like. And these today are known as amyloid plaques. In this photo taken from Wikipedia, which is a more modern section in staining, we can see these brown blobs here, which are the amyloid plaques. So, to give a very short history of amyloid in Alzheimer's disease, we can say that in 1906, Alois Alzheimer describes what we now call amyloid plaques and neurofibrillary tangles in the brain of someone with senile dementia. And then in 1984, Glenner and Wong, and in 1985, Masters and colleagues identified by sequencing the amyloid beta peptide. So they found this small protein fragment called the amyloid beta peptide, which came from these blobs in the brain that stain in a starch-like manner. Using this knowledge of the sequence, in 1987, 4 research groups identified the gene and therefore the protein from which the amyloid beta peptide is derived. And we now know that as the amyloid beta A4 precursor protein gene, APP, because the amyloid beta peptide is actually something that is cleaved out of a much larger protein, a precursor protein. Then it was discovered that the APP gene resides on chromosome 21, which made sense because people with the condition trisomy 21 tend to get early onset Alzheimer's disease. And also it was found that mutations causing familial early onset Alzheimer's disease occur in the APP gene. And this led to the formulation of the amyloid cascade hypothesis in 1992 by Hardy and Higgins. And since then, we've been stuck with amyloid beta as the center of the Alzheimer's disease research universe. And that means that most people regard, or most of the researchers in the field regard amyloid as causative of the disease, as the main pathological driver, as we could call it, although there are people who disagree with that, including myself. Now, let's look at this amyloid precursor protein. Here's a paper from only a few years ago, 2022, in Molecular Neurobiology by Delport and Hewer. And it's basically a review regarding research on the amyloid precursor protein. And I'm referring to this paper because I like this figure from the paper where they show diagrams of the amyloid precursor protein. Now there are 3 different diagrams here because actually there are 3 different versions of the amyloid precursor protein. These are what we would call isoforms. They contain similar structures, of course, but also differences. And it turns out that the form that is found mostly in the brain is this form, the APP695 form. That means it has 695 amino acids in the protein, whereas these are larger forms because they include additional parts. Now, the way that this can happen, that you get 3 different proteins from one particular gene, is through something called alternative messenger RNA splicing. So the gene has one set of information, but then in the process of making the protein from that gene, some of that information can be excluded in order to make, for example, a smaller protein such as this. They're both shown as having the same length, but we can see that there are there are certain areas missing. Now let's focus on this APP695 form and look at it a little bit more closely. In particular, I want you to just notice down here that they're marking a region called amyloid beta. So I've taken the APP695 form and I flipped it on its side, and you can see here that they're indicating the lipid bilayer. This will be a membrane of the cell, and you can see that the black bar here indicates the region of the amyloid beta A4 precursor protein that is ultimately becoming the amyloid beta peptide. So it's just this region here that actually ultimately will become A beta. Now let's focus in on this particular A beta region here. We can show this A beta region in the context of the entire protein here in another diagram. This is actually a diagram I drew for another paper. You can actually find this diagram in that review type hypothesis paper that I've written about the familial Alzheimer's disease genes. So we have this amyloid beta peptide region of the APP gene that, as you can see, is derived from the part of the APP protein that is normally mainly residing within the lipid bilayer, the cell membrane here. And that's shown here too, with there's the lipid bilayer and here's the A beta region here. And what I'm showing on this particular diagram is a number of the mutations that cause familial Alzheimer's disease and the age at which they would tend to cause the disease. So down here we have what's called a mean of mean onset ages. So this is derived from a number of studies or whatever. And what we can see here is the amyloid beta region spread out in detail to show the actual amino acids from the amyloid beta peptide. It tends to be 40 to 42 amino acid residues long. And the red dots indicate the position of particular mutations, for example, and you could say how serious they are, because how early they tend to cause the disease, because the earlier they tend to cause the disease can be regarded as a measure of the seriousness or the severity of the mutation. What you can see is that the most severe mutations occur in this particular region, but there are also mutations elsewhere. Also over here are indicated examples of where the actual protein coding sequence of the gene, the protein in other words, is not changed, but what's changed is that there are additional copies of the gene. So we call this a gene duplication. So just having too many copies of this particular gene can cause familial Alzheimer's disease. And I've already referred to one example of that, people who suffer from trisomy 21, which is having 3 copies of chromosome 21, on which the APP gene actually resides, those people tend to get early onset Alzheimer's disease. So those cases, that information is listed down here on the side. But for the ones here, this is the particular amino acid changes that cause the disease. And interestingly, as you can see, here is one of the places where the APP protein needs to get cut or cleaved, as we say. And if we look at this particular place, we can see that there are a number of mutations in that place, and presumably because most mutations usually reduce the functionality of something, we can assume that most of these mutations are reducing the ability of the APP gene to be cut in this particular position. And we know that's the case from various studies that have been done by people. Okay, and I'm also just showing you in this diagram, once again referring to the fact that it's this peptide that's found in these amyloid plaques. Here's a page from that paper I referred to earlier, from 1992, the Hardy and Higgins paper that first defined the amyloid cascade hypothesis. And the reason I want to show you this particular paper is because, as you can see, if this is a diagram here of the amyloid precursor protein, they've got it named there, this particular stroke here with 717 next to it refers to a particular mutation that you could have seen on the previous slide, which is occurring in this region where the peptide needs to be cut out of the precursor protein. And it was because some of these mutations at least were near the places where the peptide needs to be cut out of the main protein that the idea came up that these mutations must somehow be promoting the formation of amyloid beta, and therefore amyloid beta is the pathogenic agent. But as I also just mentioned to you, the trouble with a lot of mutations, the majority of mutations, if they're going to do anything, they're going to decrease a function. And in this case, the mutations at this particular position in the amyloid precursor protein that needs to be cut to produce the peptide, those mutations actually reduce the tendency of the protein to be cut. So even though many people say that this paper is the one that led to the current focus on amyloid, as it were, that's not actually fair, because even in this particular paper, they do look at the evidence in an objective way, and they basically aren't able to very strongly support amyloid itself as the pathogenic agent. What they do say, and keep in mind here that they call A beta or amyloid, the peptide, they call it A beta P here. They do say that our cascade hypothesis states that A beta P itself or APP cleavage products containing A beta P are neurotoxic. So they're not precisely focusing on A beta, and they're saying that products containing A beta are neurotoxic. Now, here's the important thing. Now, in order to be cleaved out of the amyloid precursor protein, the amyloid beta peptide needs to be cut here, which is called the beta site, and it needs to be cut here, as I've mentioned before, which is called the gamma site. And if you only cut at the beta site, you form a fragment here, which is basically mainly the transmembrane part of the APP protein, plus the intracellular part, the part that's actually inside the cell. Now, this is known as the beta C-terminal fragment, beta CTF, also known as C99, because it's the C-terminal. It involves what's called the C-terminal of the protein, because proteins are defined as either having an N-terminal or a C-terminal. and it's 99 amino acid residues long. But I think the name beta CTF is better. At the moment, these two names are bouncing around in the Alzheimer's field and possibly causing some confusion. But anyway, what you can see is that this here is actually a fragment of APP containing the amyloid beta region. And so even in their amyloid cascade hypothesis, Hardy and Higgins didn't exclude that this actually might be the pathogenic agent that's driving the disease. And that actually, I think, is what is happening. And I think there is more and more evidence accumulating supporting that. So that's a very brief introduction to the APP gene and a little bit of the history behind it. There are two other main genes that are thought of as being familial Alzheimer's disease genes, presenilin 1 and presenilin 2, and we'll talk about those in the future. And how do the mutations in all these three genes cause Alzheimer's disease? Well, hopefully I'll be able to draw the picture together for you ultimately. It's something I've spoken about in this paper that I keep referring to, which is quite technical, but in the future we'll be able to look at some of the ideas in that and hopefully get an understanding, as much as at least we have, about this mystery of how the mutations in the three familial Alzheimer's disease genes cause the disease. See you.